Good morning, I'm Rudi Agius from the Biomolecular Modeling Lab at the London Research Institute at Cancer Research UK. So today I'll be talking about how we can characterize changes in the rate of protein-protein dissociation upon interface mutation, and I'll show you how we can do this using hotspot energies and organization. So to start off, I would like to give you, just to put everything in the context, the role of off-rates in drug design. So for example, in COPD, which is basically a collection of pulmonary diseases that are characterized by poor airflow, there are two main drugs of choice here. So ipratropium and tiotropium bromide. So both of these, the drug mechanism is that they bind to the Mg muscarinic receptors, which leads to a reduction in smooth muscle contraction and opens up the airways. Now, what is interesting about these two drugs is that they have very similar structures and very similar pharmacokinetic profiles. But the difference is that tiotropium has a duration of action of 24 hours, which makes it the drug of choice because it can be administered once a day. The key thing here is that this difference in the duration of action of the drugs is down to the rate of dissociation, where tiotropium has a higher residence time. Basically, the residence time can be calculated as 1 over the off rate. So there are several other examples where the long duration of action is related to slow off rates. The key thing here is that generally kinetics are only investigated very late in the drug design lead optimization scenario. So the problem there is that you might have compounds which are though slightly less potent, but they may have better kinetic profiles, which would be discarded. And obviously the problem is that experimental methods so far, they have a very high cost per data point acquisition. So this sets the stage very nicely for having computational prediction of off rates and changes in off rates upon mutation. So the idea is that given a structure of a receptor-ligand complex, we want to be able to predict the off rate of the wild-type complex, and for a number of mutations, we want to be able to predict the changes in the off rate. From that, we can calculate the change in K off. Now, the key thing is that from these mutations, we want two things. Ideally, we want to be able to rank them accurately according to delta K off. And getting back to the drug design scenario, we are very interested in mutations which can stabilize the complex, which can increase the residence time. So we will concentrate mainly on these two goals, at least for this talk. So as I said, I'll be talking about ranking mutations. So these mutations can be to any residue type and also multi-point mutations. And I'll show you how we can also detect these rare mutations which can increase the residence time. Hopefully, I'll show you how we can do this using computational alanine scans. And from these alanine scans, we can generate what are known as hotspot descriptors and use these descriptors to characterize the changes in off rate. And we benchmark this on a set of 713 mutations on 50 different complexes for which we have experimental off rates calculated for. So to start off, a good place to start off is looking at the binding mechanism process. So for example, if you assume that conformational changes are minimal, let's say, then the K off is simply the kinetic parameter K2. Whereas if you have conformational changes, then the K off changes, and you would also need to consider not only the stability of the bound complex, but also the stability of the transition complex. So in this work, we assume a one-step binding process because most of our structures that we use do not undergo significant conformational changes. So the quick question that we want to ask really, and a kind of a naive question, is what makes complexes stick together? Remember, we want to calculate some likelihood of how easy it is to get from this bound state to the unbound state. And how can we quantify the stickiness? So I will show you the way we try to do it, maybe less of a traditional way, but hopefully I'll show you that it's worth it in the end. So just think of this as a toy representation of the interface. So these can be residues from both the receptor and both the ligand. So what do we know about complex interfaces? Well, alanine scanning gives us a lot of information on the interface. So in alanine scanning, basically, you turn each residue into alanine mutation, and you can calculate the change in binding free energy, for example, which is the delta delta G. In this case, if it's a positive delta delta G, then it's destabilizing. So that mutation destabilizes the complex by increasing the binding free energy. So if you start scanning the interface one by one, you would find that certain hotspots are able to destabilize the complex more. And we term these as hotspot residues. And so then, at the end of a, let's say, a lanolin scan, you can have a nice representation of the interface with hotspot residues and non-hotspot residues. Now, work on hotspots can be categorized into, let's say, 3 scenarios. So first, we have quite a good number of computational hotspot prediction algorithms. The problem is that we don't have a lot of applications so far for them. So one, for example, interesting application that we have is that hotspots serve as very good druggable sites in protein-protein interfaces. And other work related to hotspot residues has concentrated also on their properties in interfaces. So for example, we know that hotspot residues tend to cluster into hot regions, which is a cluster of hotspot residues, and they tend to occur also in the central parts of the interface, which we term as the core region. But what we wanted to do is we wanted to make that intuitive link between the number of hotspots or the hotspot energy and the stability of the interface. So the idea is, very naively, are interactions which have more hotspot residues, are they more stable? Do they have a lower off rate? So the hypothesis that we set up is that seeing the hotspot residues as anchor points of a complex, then a mutation which increases the energy contribution of hotspot residues should maybe result in a more stable complex. So for the wild type, how we can test for this, for the wild type interface, we make a computational line scan, predict the hotspots, get the delta delta Gs, and sum the contribution of these hotspots. Now remember, this is a protein complex for which we want to apply a mutation to and predict the change in off rate. So let's say this is a lysine to leucine mutation on this residue. And we apply the mutation, we rescan the interface for hotspots, and let's say this mutation realized another new hotspot. So the idea is that you sum the contributions again, and in this case it's 14.2 kcal per mole. So the idea is the mutant complex has a higher hotspot energy contribution than the wild type complex, and according to our hypothesis, then this mutant interface would be more stable through a lower K-off and a higher residence time. So just putting things in terms of equations, this is one of the descriptors that we use, which is the change in interface hotspot energies, which, as you can see, sums the delta delta Gs of the hotspot residues for the mutant minus the hotspot residues for the wild type. And we want to check whether this correlates with change in K-off. So to do so, we used a SCMP database. I will explain in a couple of slides later, but just for now, you should know that we have a list of experimental delta K-offs. And what we do for each complex and mutation, we do the method that I showed you before. So we can calculate the delta and THS energy. And to check how well this can correlate to change in K-off, we calculate the Pearson correlation coefficient. And we find a correlation coefficient of minus 0.56. So this is interesting because we have a reasonable correlation, so there is some evidence that this might hold. And the negative sign implies that an increase in the hotspot energies results in a decrease in the off rate. So confirming this hypothesis, then what we go on to do is we design a set of 16 hotspot descriptors. I will not go into them, but basically the idea of these hotspot descriptors is we could test for several things related to the distribution. For example, we could test whether large hot regions make a complex more stable, etc., etc. Now, what we do, we want to benchmark these hotspot descriptors to more traditional descriptors. For example, descriptors such as the hydrogen bonding or the van der Waals potential or surface complementarity. So we have a nice broad set of molecular descriptors which we can compare. And again, we want to check the ranking ability, we use Pearson correlation coefficients, and we also want to detect, most importantly, those mutations which increase the residence time. And in the end, I also show you some machine learning models which combine both descriptors. So starting off with comparing how well we can accurately rank mutations. Okay, so I'll explain quickly the data set. So we use ISCB, so this is a very large database. It has around 3,000 mutations with binding free energies, entropies, enthalpies, and off rates also. So from this, we extract 50 complexes which have 713 mutations with delta K off. So we have a nice regression data set there. Since we're also interested in these mutations, which can increase the residence times, we also create two classification data sets. So this is a histogram of the range of mutations you have. So for example, one should note that the stabilizing mutations, the ones that increase the residence time, are very rare. And so we just have 31 out of 713, and we concentrate a lot on the detection of these. So we create two data sets, one CDS1, which has positive examples and negative examples, where the negative examples are simply neutral to destabilizing mutations and the positive are stabilizing mutations. And we take another data set which has stabilizing examples and destabilizing. So you remove the neutral point. You make it a little bit easier, basically. Okay, so first off, we want to check how accurately we can rank mutations according to K-off. So we compare the hotspot descriptors to the molecular descriptors I told you before. So what you see here is, so in blue, you can see a rank. So these are all different molecular descriptors, and they're ranked according to their absolute PCC to the K-off. So it shows how well they can characterize changes in the off rate. And in green, you can see the hotspot descriptors. So immediately, you can appreciate that these hotspot descriptors are consistently better at characterizing the off rates upon mutation. If we zoom in a little bit on the top 10 descriptors, we can see that molecular descriptors are dominated by statistical potentials, and the hotspot descriptors are dominated by the interface hotspot energy that I showed you before. You can see multiple instances of interface hotspot energy is because we test a number of computational hotspot predictors just to validate the method. So if we take a closer look at what the scatter plots would look like for the best molecular descriptor and the best hotspot descriptor, you can see that the destabilizing portion of the data set, we can do pretty well in that. But where we really fail is predicting those stabilizing mutations. And so as you can see, we have quite a number of false negatives and false positives. So another thing that we check is, again, we are very interested in these mutations which increase the residence time, those rare mutations. So we use the classifier data sets now and calculate a number of the MCC and U-test and a bunch of other classification-related measures. So again, what you can appreciate here is that in a ranked list, the hotspot descriptors in green tend to dominate. And similarly, if we check for the second classifier data set, which removes the neutrals, again, the hotspot descriptors tend to dominate. So just to give you an idea of what we're looking at in terms of detection rates, so the top molecular descriptor here, which is a docking-related potential, it has managed to just to detect 14 out of those residence time increasing mutations, whereas the top hotspot descriptor manages to detect 18 of these. Keep in mind that it does this with a lower or similar false positive rate. Okay, so as a summary of the first part, we can see that using computation-aligned scans, the changes in energies of hotspot residues pre- and post-mutation can be used to estimate a change in the off rate. Secondly, we've seen that hotspot descriptors can capture changes in the off rate consistently better than benchmark set of molecular descriptors, which includes a broad range of potentials and biophysical descriptors. And thirdly, we see that we can detect more mutations which increase the residence time with hotspot descriptors. Now, the next thing that we do is obviously we want to improve. Remember, these are results based on just a single descriptor. So what we do is combine these descriptors into machine learning models to try and achieve even higher performance. So I will not go through this. I'll go through this quickly. So we have the same data sets, regression and two classifier data sets. We have the descriptors, which I talked about. We feed them into random forest regression and classification models. And using cross-validation, we check how well we do in the prediction of K-offs. So what I'm showing you here is the scatter plot of an off-rate prediction model trained solely on molecular descriptors only. This is a model trained on hotspot descriptors only, and this is a model trained on both on both hotspot and molecular descriptors. So what's nice is we can characterize the destabilizing portion very nice. We still tend to underestimate somewhat the stabilizing portion, but at least we have much less false negatives and false positives. And as you can see, the false negatives increase from moving from a molecular descriptor model to adding hotspots. But to check this exactly, we also, again, do classification models, so we can check how well we can do in this. So again, we train some classifier models on CDS1, and we see that we get more detections for the hotspot models, and similarly for CDS2, which removes the neutrals, we get even higher detection rates. So if we concentrate a little bit on these 31 residence time increasing mutations, which are listed here, so what we can do is, for all the models that we tested, we can check the hit rate, so the number of detections. So for example, as you can see here, the mutations here basically were detected by all descriptors, whereas this by all off-rate models, whereas this multi-point mutation was not. So you can see these are kind of harder to detect, let's say, than these ones. And why is this so? So when we look at these, we find that on the harder portion, it's usually mutations to alanine. So this is interesting because these are mutations to an alanine residue, which actually stabilize the complex. And this is where we kind of fail. We fail at detecting these quite as well as other types of mutations. So in the second part of the talk, we've seen how we can accurately rank mutations according to delta K of using a number of hotspot and molecular descriptor models. So the highest we achieve is a 0.79 correlation coefficient. We've seen that residence time increasing mutations tend to be undetected or underestimated using molecular descriptors, but this is elevated when using hotspot descriptors. And as I told you earlier, residence time increasing mutations which involve mutations to alanine residue are the hardest to detect. As one final thing I wanted to talk about is something related to the distribution. So for example, we've seen so far that counting the energies of hotspot energies, pre and post mutation, provides, let's say, an accurate, reasonable description of changes in delta K of. Now let's think of, let's say, a drug design scenario. So, okay, you've made a computational align scan of the interface. You know that these are good if you want a stable interface, these hotspots, but you want to stabilize it further. So, okay, we know that if we increase the hotspot energy or somehow add new hotspots, we can stabilize it further. But the question is, you have so many options here. So are there certain positions where we can actually gain an advantage by putting hotspots there? For example, if we add an hotspot here, so make that hot region larger, does it help better than just adding a hotspot here? So we wanted to kind of probe a little bit whether the distribution of hotspots actually matters. So to do this, we know two common distributional properties related to hotspots. So we know that they preferentially occur in the core region of the complex. So this is related to the Oring Hypothesis. So the idea is that the important parts of the complex are centralized towards the core, and rim residues, they tend to play a more of a secondary role. So they're just there to shield the solvent from the core region. So what does this mean in terms of stabilities? For example, does it mean that if we have mutations which affect core hotspots, can we affect K of stabilize or destabilize K of more significantly than rim mutation hotspots? So that's one thing we wanted to check. Another thing that we know is that hotspots tend to cluster into tightly packed regions. So again, the larger hot regions so imply a more stable interface. Since I do not have time to go into both of them, I'll just talk briefly about this. So just to remind you, I haven't shown you this descriptor, but as I told you, I've designed a number of descriptors. So the descriptor that I showed you before was interface hotspot energy. So the descriptor that I described before calculates the change in the total sum of hotspot energies pre and post mutation. What core hotspot energy does is it forgets about what happens at all the parts of the interface except in the core region. So the idea is that if we try and estimate changes in the off rate of any mutation by considering only what happens in the core, so forgetting totally what happens in other regions, but you still succeed, you still can actually characterize a change in off rate by just considering what happens here, then the core hotspot energies seem to be important for complex stability. Similarly, if we look at rim hotspots, so rim hotspots are the outer region of the complex. Similarly, we can say if you try and estimate changes in off rate by considering only what happens in the rim, forgetting all other places, but you can still actually correlate that with a change in off rate, then they are important for complex stability. So to check this, from the 713 off rate mutation dataset, we see which mutations affect core regions and which mutations affect 148 regions, and we check the correlations with the off rate in these two instances. And what we find is that changes in core regions are more indicative of changes in the off rate, whereas for rim region mutations, it's not so. So this is very kind of in line with what the Oring hypothesis suggests. So we wanted to probe this further. So what if we change the interface area of the complex or change the size of the complex? Do the rules change? So what we do is divide the data set into small interface area and large interface area, and we do the same thing. We check whether mutations in core hotspot energies can correlate to KF, and similarly for rim region mutations. So what happens is that as you move from large interface area to small interface area, rim region mutations start to play a more primary role. If we do the same thing with complex sizes. When we move from large complex sizes to small complex sizes, again, rim regions start to be a little bit more important, at least than what they were before. But still here, we have some confounding factors, because we may have small interface areas which have small complex sizes or large complex sizes. So what we do is further divide the dataset into all possible scenarios. And we do, again, the same thing. So looking here, just fixing for interface area, so fixing for small interface areas, and then varying complex size, we find that for large complex sizes moving to small complex sizes, rim regions show the same thing. They become more primary. And similarly, if we fix for large interface areas, again, when we move from large complex sizes to small complex sizes, we see a greater role in the rim regions. So what we are seeing here effectively is independent of interface area, it is complex size that affects the role rim regions play. And this is summarized here. So the idea is that when you want to apply mutations and maybe stabilize the complex, depends what size complex you have, you may be better off mutating, for example, in the rim regions rather than the core regions, or at least both of them. So just to close off, other things that we have investigated, but I have no time to talk about here, is that, first of all, the number of hotspots, just looking at the counts of hotspots, is not enough to characterize changes in off rate. So you need to look at the energies of them, the change in the sum of energies. We also look into different cooperative effects between hotspot residues. So if you assume positive cooperativity between hot regions, it's not necessarily that having more hotspots increases the stability, because you reach a saturation point. What we found is that the prediction accuracy significantly degrades for complexes which undergo conformational changes, but to a certain extent this is expected because we are only assuming a one-step complex dissociation model, and for that we need two-step complex dissociation model, and we need to also characterize the transition state, but that is not trivial. And this is something that training models, if we train models instead of the whole data set, for example, instead of the whole 730 mutations, we train models specifically to regions, to core region mutations, to rim region mutations, we can improve prediction accuracy. And as just current work, what I'm currently working on now is I'm trying to use that same idea, so making computational align scans of true interfaces, true complexes, and false positive docked poses to try and see whether hotspots and their prediction that their distribution can discriminate between true interfaces and false positives. So with that in mind, I would like to thank you for your time and thank my lab and my collaborators.
Hi, thanks for the wonderful talk. So it's more of a technical question. You had a classification problem essentially, in the way you decided that it would be a classification problem was you set an energy threshold. You had a greater than 1 or less than -1 threshold for the
Exactly.
energy, right? I'm aware that there are different thresholds in the literature. Is there any reason why you chose that? And what were the effects if you tried other thresholds?
So, okay, so for starters, no, there are no rules on how we set the threshold. However, the idea is that let's say if you look at these mutations, okay, it's negative. So these should be stabilizing mutations, but they're really so close to zero that you can't really consider them as stabilizing. So it's kind of safer to assume that they're neutral or discarding them. So that was basically the idea to choose those thresholds. And similarly for the next data set, we wanted to increase this threshold so that we can see kind of a clear distinction between destabilizing and stabilizing. So seeing if that actually helps the prediction. But in reality, the scenario, in reality, it would be even without this threshold. That's what you would have. Okay, so what we do, so for the prediction of delta G's, as I said, we can use hotspot predictor algorithms. So what we do is we develop our own hotspot predictor algorithm, and we also compare to currently available hotspot predictor algorithms. What I found is that we can do it very accurately for mutations which tend to occur in the core region of the interface. Whereas if you go outside towards the rim regions of the interface, the accuracy degrades, and it's pretty consistent for most of them.
Okay, so let's thank our speaker again.
Thank you.